And now we are recording. So today we're doing brains. We're going to do organization of the nervous system, cytology of nervous tissue. This will be review from your tissues lecture. Myelination of axons. There's going to be a handful more cells types amongst your glia. You might remember from your tissues lecture that you have neurons and glia in nervous tissue. Turns out there's a bunch of different kinds of glia that we're going to need to learn about 6. Axon regeneration, or lack thereof. When can axons regenerate? When can they not, and why? Nerves, synapses, and the action potential. Neural integration, neural pools, and development of the nervous system. Not as key to synapses and the action potential. That's the key concept. There are a lot of ways that we classify and organize the nervous system. We can do structural or we can do functional. Either way, it's only one nervous system. We can divide the nervous system into the central nervous system, or CNS, and peripheral nervous system, or PNS. And yes, I have to enunciate PNS for obvious reasons. The central nervous system includes your brain and spinal cord. Get this going. So brain and spinal cord. That's a key concept. You have to recall for all time that your spinal cord is considered part of your central nervous system. There is going to be clinical relevance to that. Your peripheral nervous system includes your cranial nerves and your spinal nerves. Feel free to ask questions at any point. Since I'm recording, I may repeat the question for the person listening since you're not as close to the mic as I am. One thing that we're going to do over our entire nervous system unit, including today and on, is we're going to start organizing things into, is this a collection of cell bodies or is this a collection of axons? We're going to talk about the anatomy of the nervous cell, the neuron cell itself. And there are cell bodies and they have these long extensions called axons. And when we say things like nerves, ganglia, we're just talking about one or the other. We're talking about, in the case of nerves, collections of axons that travel together, things that travel long distances, whereas ganglia are going to be collections of cell bodies. And we'll deal with that a little bit more later. We'll start our official cell body versus axon list a little bit later. Again, with the recording, it's hard to do in this format. The role of the nervous system is to collect information, process it, and then send information out. This is the same model as our homeostasis model. Homeostasis means that there is a sensor somewhere in the body that senses the internal state or external state of the body, like temperature, hunger, thirst. And that sends that information to a control center. And most of the time that control center is going to be the brain. Or I should say the CNS. We're going to see more CNS stuff. Then it's going to send out an effector. For example, our motor neuron that we now know a little bit about. A motor neuron is an example of an effector, something that's going to change your status, either from, like, say you shiver to keep warm, or whatever it is. So your your nervous system is an excellent way to participate in those homeostatic mechanisms. It's going to control everything. Here's your first your functional organization of the nervous system. You can divide it into sensory and motor divisions. Sensors send information to the central nervous system. Motor nervous system sends out information along effectors, things that are going to change your state. We have further subdivisions. We have somatic sensory and visceral sensory. And in the motor nervous system, we have somatic motor and autonomic motor. So back over here on sensory. Somatic sensory means sensory information we're consciously aware of. Skin, joints, skeletal muscles, special senses like sight, sound, etc. We're consciously aware of these things, and the things that we do about them could be conscious or unconscious. Visceral, again, refers to viscera. If you have a gash on your small intestine, are you going to be able to point to the gash on your small intestine from the outside and say, here's where my gash is, go in and fix it? Absolutely not. It's vague. So most of your visceral sensory information is going to be unconscious, and not all of it is even going to be physical sensory. You have completely unconscious knowledge of things like your blood pressure, your chemical state, your electrolyte balance, things you've probably never really had to think about that much before. Any questions so far? This is going to be on your worksheet today. It's literally this one. I'm emphasizing this because jazz hands, you've got to know these divisions. Motor nervous system, you have somatic and autonomic, and it's going to cover a similar range as we did in our sensory system. Somatic motor generally is going to be voluntary, things you can do consciously. Innervating the skeletal muscle, allowing you to move and dance and take notes or whatever it is. Autonomic motor is going to be very much like visceral sensory, where it's involuntary and or unconscious. Cardiac muscle, smooth muscle glands, you are not consciously aware of what your pineal gland is releasing. You're not consciously aware of what your pancreas is doing. If you had to be conscious for those things, it would be very difficult for you. But thank goodness that's on automatic. As far as I'm concerned, when you see the word autonomic, you can go ahead and say automatic. Again, you still need to write the word autonomic or recognize the word autonomic, but it does have that connotation of automatic. And this is going back to this concept of visceral versus parietal, something we've been doing since day one. Parietal means body wall, viscera refer to internal organs. This is a concept that's going to come back in a couple of different ways and a couple of different times. One of the reasons that we have that vague, or actually I should say, one of the reasons there's fallout from having vague innervation of internal organs is that sometimes we get referred pain patterns from that. You have what we're going to later learn are called dermatomes, areas of skin, your body wall, your parietal innervation, that are very highly organized, especially over your trunk. And deep to those structures, you have your viscera. And those nerves from your viscera are not going to be in the exact same territories as your dermatomes. So for example, your appendix may be over here somewhere. Can you actually see? Yeah, you can. Okay. Your vermiform appendix may be over here somewhere. But if your vermiform appendix is infected, you may feel it over here in the umbilical region. That's a fairly well-known referred pain pattern. And the reason is that the nerve, the visceral nerve that serves the appendix, is ascending to this level before it joins up with the dermatome for T10, and that goes to your spinal nerve, and that goes to your spinal cord. And in the spinal cord, there can be crosstalk. We'll have two synaptic knobs right next to each other, and the neurotransmitter from one can diffuse over to the receptor for another one. That's what a referred pain pattern is. And that's a concept that's really more for another day, but I'm just trying to highlight that difference between visceral versus parietal innervation, where parietal is going to be very conscientious, and visceral is going to be a lot less specific, unless you've got a referred pain pattern, and in which case you can get some fairly reliable patterns with a lot of exceptions to the rule, like me. My appendicitis did not look like that. Okay, we did that already. Questions so far? So far so good? Or with me? It's making sense? You've already seen the histology of nervous tissue before. The neurons are going to be several times larger than the glia. The glia are going to be all of these little cell bodies out here. The nerve cells are electrically excitable, much like your muscle cells are electrically excitable. Those are going to be very much related concepts. Whereas your glia, glial, glia plural, non-excitable cells that support and protect neurons. And there's going to be a number of different types of glia, and we're going to learn their names, and we're going to become friends with them because they're pretty awesome. Because neurons are electrically excitable, because they are making neurotransmitters that are, for the most part, proteins, not all of them are, but most of them, they have a high metabolic rate. A lot of the glucose, a lot of the nutrients that you take in from food, a disproportionate amount is going to your brain to fuel your nervous activities. They have extreme longevity, which means you're actually born with most of your neurons. In the adult, most neurons do not undergo mitosis. So it's similar to your muscle cells, where if you get damage to your brain tissue, to your central nervous system, it is unlikely to heal itself as brain tissue. Yes?
So how is it that people who have had an accident and go like quadriplegic, that they can regain?
It depends on a lot of factors, and we can talk about that quite a bit later on. Yeah, we'll talk about Wallerian degeneration and how that gets cut down. And I've written an entire paper on this before, so we can go into it. Non-mitotic. There is actually one brain region where it undergoes mitosis, but it is a weird exception to the rule. So again, if you just had a baby, it's already got all the neurons it's ever going to have. Maybe we'll get a few more, like during college. College age, you tend to peak with your neuron count, and then it's all downhill from there. I'm quite serious. It's all downhill from there. So the structure of your neuron, ignore all the extra lines. I think, this picture or a similar picture on my worksheet today. And all I really want you to label is the cell body, the axon, and the dendrites. So the cell body's up here. That's where the nucleus is. And it's going to be surrounded by a bunch of processes that we're going to call dendrites now. It looks like the branches of a tree. Those are the dendrites. That's the receiving end. This end of the neuron is going to receive information. And the nucleus is going to process information. And that axon is going to send information.
The dendrites receive information?
Correct.
The body processes it?
Correct.
And then what are the axons?
Send information. And what you're looking at here is an extreme zoomed out segment of multiple synaptic knobs. These are the same synaptic knobs that we know from our muscles lecture. One axon, typically there's only one axon. You should, in most neurons, find only one axon. But that axon can split into multiple synaptic knobs. I'm going to start calling them axon terminals. Axon terminal. Terminal of the axon, the end of the axon, right? So back here at the dendrite, the beginning, the cell body processes, the axon sends along, the axon terminals are ending. This is where those neurotransmitters come flying out of that neuron. Yep. This is a nice cartoony version with our dendrites looking nice and cartoony. Those dendrites are going to have a number of spines on them. We're going to call them dendritic spines. Those are going to participate in neuron-to-neuron synapses. So what's going to happen is one axon is going to come down here. That axon terminal is going to connect to one of these dendritic spines on the dendrite. And that's where you're going to have your neuron-to-neuron synapse. You're going to have neuron-to-neuron communication this way. That's why it's kind of unidirectional flow. We have one neuron releasing information onto another neuron, and that neuron takes that information, processes, and sends its own information along. It's a chain. It's a link. It's like a game of telephone, but a little bit more reliable. Not a lot more reliable. Your brain is not reliable, unfortunately. Questions so far? So we have our typical cell body and we have our neuron cell body, and actually they're pretty similar. You've got your same old structures. You've got your nucleus, you've got your endoplasmic reticulum, you've got mitochondria. You should have a lot of mitochondria because it is energetically expensive to make all of this neurotransmitter and to fire all of these action potentials that we're going to talk about. Lots of ribosomes because, again, most of our neurotransmitters